Transcription factors are the master regulators of our cells. These specialized proteins act as molecular switches that control the expression of genes by binding directly to specific DNA sequences. By binding to DNA, transcription factors can initiate the process of transcription, turning genes on, or they can block the cellular machinery, effectively turning genes off. This precise control determines cellular identity. A neuron and a muscle cell contain the same DNA, but different transcription factors dictate their unique functions by governing the genetic program. Development is governed by hierarchical gene regulatory networks. Think of these as a biological circuit board. In this system, the output of one gene acts as a vital input for another, establishing a complex chain of command. This logic creates a cascading program, systematically driving the precise formation of tissues and organs. Every cell in your body contains the same complete DNA sequence, acting like a library of thousands of books. However, not every cell needs to read every book. Epigenetic switches are the locking mechanisms of the genome. They consist of chemical markers that bind to DNA, determining which genes remain open and accessible. When these markers attach, they condense the DNA structure, effectively locking the gene and preventing the cell from reading that specific instruction. This ensures functional stability. A liver cell uses epigenetic markers to lock away the genes for a neuron, preventing accidental expression of non-native biological programs. By orchestrating this complex lock-and-key system, the cell maintains its specialized identity throughout the organism's entire life cycle. Cell fate specification is the developmental journey where a versatile, pluripotent cell commits to a specific lineage. Initially, a stem cell possesses the potential to become any cell type in the body. As development progresses, the cell follows a specific genetic program, gradually restricting its options. Each branch point represents a commitment step where the cell permanently locks away alternative fates. Once specified, the cell is destined to become a specialized component of a specific tissue. This process ensures that complex structures like organs and systems are built from precisely defined functional units. Hox genes are the master architects of biological form, acting as the global positioning system for the developing embryo. They define the identity of segments along the head-to-tail axis, ensuring that organs and limbs develop in their precise anatomical locations. These genes are arranged in clusters on the chromosome, mirroring their order of expression in the organism, a phenomenon known as collinearity. As the embryo matures, Hox genes activate sequentially. Each gene provides a unique chemical address, telling a cluster of cells exactly where they are and what they should become. Without these genetic instructions, developmental chaos would ensue, misplacing structures and failing to organize the body correctly. Hox genes are architectural master switches. Their physical order on the chromosome, a phenomenon known as collinearity, mirrors their precise order of expression along the developing body axis. This spatial arrangement is strictly maintained. As the embryo matures, the sequence of gene activation translates directly into the structural identity of segments from the head down to the abdomen. This genetic blueprint is remarkably conserved. From fruit flies to humans, the same fundamental Hox gene clusters guide body patterning, showcasing a deep, ancient evolutionary link spanning hundreds of millions of years. Traditionally, to sequence gene activity, we had to grind up tissue samples. This destroys the physical context, losing the vital spatial information. Spatial transcriptomics changes this by allowing us to identify gene expression while preserving the tissue architecture in 3 dimensions. By mapping molecular signals back to specific coordinates, we generate high-definition maps of where genes are active in a living embryo. This breakthrough empowers us to see development not just as a sequence of chemical reactions, but as a precise spatial execution of genetic programs. We are entering a new era of developmental biology where artificial intelligence allows us to build digital embryos. These computational models simulate how complex gene regulatory networks function in 4 dimensions. By adjusting genetic parameters within the AI, we can observe how specific mutations alter the physical form of the developing organism. This predictive power allows researchers to forecast developmental outcomes with high accuracy before ever setting foot in a laboratory. Developmental biology has evolved into a study of biological computation. We now view the embryo not merely as a growing mass, but as a sophisticated living computer. Just as transistors execute logic gates, cells process chemical inputs. When morphogen signals cross a specific threshold, a logic gate triggers the expression of downstream genes. This creates a cascading network. The embryo computes its own physical form, transforming simple genetic code into a complex functional organism through successive computational steps. The complexity of an organism is not determined by the number of protein-coding genes it possesses. Instead, it lies hidden within the vast expanses of the genome, once dismissed as dark matter. Non-coding DNA acts as a sophisticated regulatory control center. These regions contain switches and enhancers that orchestrate the precise timing and expression of genes during development. As species evolved, their non-coding regulatory networks became increasingly elaborate. It is this layer of biological computation that allows for the construction of complex structures from the same basic genetic toolkit. Organoid models represent a breakthrough in developmental biology. We begin with a cluster of pluripotent stem cells, which possess the unique ability to differentiate into any cell type found in the human body. Through controlled chemical cues, these cells self-organize into 3-dimensional structures. They replicate the architecture of real organs at a miniature scale, allowing us to observe developmental milestones in real time. These models are critical for disease research. By introducing specific genetic mutations, we can observe precisely how errors disrupt organ formation, such as the folding of the cerebral cortex or the vascularization of heart tissue. These structures provide a high-resolution window into human development. By integrating spatial transcriptomics with organoid culture, we are mapping the genetic trajectories of organ maturation with unprecedented clarity. The axolotl, a unique salamander, possesses a remarkable ability to completely regrow lost limbs throughout its entire life. Researchers are currently mapping the complex genetic networks responsible for this feat. At the site of an injury, specialized cells revert to a pluripotent state, forming a structure called the blastema. This cluster acts as a localized engine for reconstruction, orchestrating the growth of bone, muscle, and nerves. By analyzing these cells, scientists have identified specific signaling pathways, such as WNT and FGF, that reawaken latent developmental programs. These are the same pathways used during initial embryonic development. This research is far from purely academic. The ultimate goal is to identify these triggers and provide a blueprint to stimulate similar repair mechanisms in human tissues, potentially revolutionizing regenerative medicine. Synthetic embryology represents a revolutionary leap in developmental biology. It involves creating embryo-like structures using stem cells, bypassing the traditional requirement for sperm and egg fertilization. By manipulating pluripotent stem cells, researchers trigger a process called self-organization. These cells autonomously assemble into complex structures that mimic the architecture of early embryos. This technology provides unprecedented access to the black box of early development. We can now study implantation and cell fate specification with high precision in a laboratory environment. Synthetic embryos are transforming our understanding of life's origins. They offer deep insights while keeping the developmental process within a controlled, observable system. Epigenetic editing allows us to control the genome without rewriting the underlying DNA sequence. Instead of cutting or pasting genes, we target the chemical markers known as epigenetic switches that dictate gene activity. By using a modified, inactive version of the CRISPR system, called dCas9, we can guide an effector protein to a specific gene location. This tool binds to the DNA without breaking the strands. Once positioned, the effector protein can remove or add these methyl tags, effectively flipping the gene's switch from off to on, or vice versa. This precision offers a way to correct developmental errors while keeping the genetic blueprint entirely intact. The ultimate goal of developmental biology is to decode the genotype to phenotype map. It is the quest to predict an organism's complex physical form solely from its raw genetic sequence. We treat development as a system of biological computation. DNA acts as the input, while the complex gene regulatory networks function as the processing engine. By mapping these dynamic processes, we move past simple observation. We are now building predictive models that can simulate the development of tissues and organs before they even manifest. This transition from descriptive biology to predictive engineering represents the new frontier, allowing us to eventually understand how life encodes complexity. Germline engineering involves making genetic changes that are inheritable. This means modifications are passed down to future generations, raising profound ethical questions. It is a delicate balance. On one side, we have the potential to eradicate debilitating hereditary diseases. On the other, we face risks of unintended mutations and the complex implications of altering our species' biological future. The scientific community emphasizes the need for rigorous safety testing and comprehensive global regulations. Before such powerful tools are applied to the human germline, there must be a firm consensus that prioritizes safety and ethical integrity. Ultimately, the power to rewrite the blueprint of life demands immense responsibility. Our choices today will shape the biological heritage of tomorrow. Developmental biology has undergone a major paradigm shift. We have moved from a purely descriptive parts list approach, where we simply identified which genes were present in the genome. We are now embracing a systems engineering approach. This focuses on how these components interact across space and time, effectively treating development as a biological computation. The modern goal is predictive modeling. By understanding the underlying logic, we can simulate developmental outcomes and predict the effects of genetic mutations long before they are tested in a physical laboratory. Regenerative medicine is entering a new era. By decoding the genetic control of development, we can now harness the body's latent programs to repair damaged tissues. At the core of this approach is cell fate specification. We can now identify the transcription factors required to reset a mature cell back to a regenerative state. This enables the potential to regenerate entire organs or correct congenital defects by guiding cells along specific lineage pathways, mimicking the embryonic process. As we transition from descriptive observation to predictive engineering, regenerative medicine promises to transform modern clinical practice. Development is an exquisite symphony of regulatory events. It begins with a single cell, governed by a precise, four-dimensional execution of a complex genetic program. By mastering the gene regulatory networks, epigenetic switches, and morphogen gradients, we are decoding the very logic of how life is constructed. We have moved from merely observing parts to simulating and re-engineering these biological processes. This shift marks the future of regenerative medicine and our understanding of biological identity.